Hey everybody, and thank you for tuning in to Chapter 10, which is all about single-subject designs. So this is I feel like a nice little break in the subject material for this semester because this one is pretty easy, I think, compared especially to Chapter 9, which is about that factorial design and about mixed models where you have, multiple layers of an independent variable. This one is much more scaled back. What I'm going to do is just going to run through this. I think probably the best course of strategy is to read ahead in the chapter first. I'm going to kind of follow along with the chapter, but I've kind of rearranged a few things and introduced a thing or two. All right, so let's get into it. So chapter 10. So I have some videos here. These videos are not really about research necessarily, but about individuals that or groups that might lend themselves well to single subject research. So what I would like for you to do is, you can do it now or you can do it at the very end of all of this, is look at each of these videos and think about in what ways could we effectively study this person's behavior in a way that would tell us about our own behavior. Okay, so let's get into the chapter. So far, I'll be honest with you. This is kind of a strange order of things. This is the first time that I'm using this textbook. Previous times, this chapter, in other textbooks, this chapter will usually come before the one that we just did. So I think a lot of this is going to sound pretty easy to you because we've gone through the harder things first. But when we talk about small n designs, the way that I see it is that there's essentially two different kinds. You got something kind of like a case study on one hand, which we've talked a little bit about this semester, and on the other hand, you have essentially an experiment with 1, 2, or 3 people. And those are the two different kinds of single subject designs we're going to be talking about. You'll hear me refer to these as small n designs. I don't know if I've talked about this that much this semester, but the letter n is oftentimes used to signify a sample size. So the n, if somebody ever asks you, oh yeah, for your experiment, what n did you have? Usually what they're just asking is what sample size did you have? And so small n just means a very small sample size design. So in experiments, usually the goal of the experiment is to get a subset of the population, right? You're going to get a sample, which is a subset of the population. You're going to test them in some way and make generalized conclusions about your findings to the broader audience. This time, we're not exactly doing that. We're going to change things up a little bit. Small n designs are typically going to be targeted for very special populations. And what I mean by that are populations that are maybe at risk, maybe vulnerable, maybe just really rare. And so because of that, it doesn't really make sense to get a subset of the population and draw your conclusions. Instead, we're looking specifically for people that meet certain criteria and then kind of work with them. So maybe we're interested in someone with a very rare neurological disorder. So for example, we'll look at an example in just a moment of someone who doesn't have a hippocampus and what we've learned from that individual. Maybe we are interested in somebody who is a famous person or a celebrity or somebody that has a unique experience. Somebody that, just as an example, so someone with a unique experience might be, let's say Sully. Sully, Sully, the captain who saved the ship over the Hudson River in New York. It was that crash landing on the river. So as far as I know, he's kind of the only person who's crash landed on the Hudson River and saved everybody. And so it might be interesting to examine his reflexes or his decision-making process or the way that he uses his attention. Things like that. You get the idea. That's a unique experience that can tell us a lot about how attention works, how reflexes work, and so on. Maybe we're interested in someone with an incredible ability. So maybe we are interested in, somebody with an extraordinary memory, or maybe somebody who is like an extraordinary athlete. Those would all be good. Or maybe we are using a few children in which we are using some kind of new technique for. So, like, let's say that we are interested in looking at speed reading. We want to teach speed reading to young kids. We would do that in a single or a small N design. Where we're working with one or two people at a time instead of trying to do an entire classroom like that. So very quickly, a couple of differences that I want to highlight between large N designs or large sample designs and small sample designs. Whenever you have a large N design, your participants are going to be grouped. Sometimes you're going to have them all in one group that have multiple conditions, but oftentimes we kind of split them out into different groups and give them different independent variables. For small N designs or single subject designs, we're going to be treating all the participants pretty much as their own individual instead of lumping them all together. In single subject designs, you don't have a control group because of that. Single subject designs are almost always going to be a within subjects design, so they're always repeated measures. In other words, people will get multiple levels of the independent variable. It's not just splitting up into groups, as I mentioned. Another thing that's kind of interesting, I think, about the differences between these two fields is that when we talk about understanding the impact of something in research methods, we usually talk about data and data analysis and using statistics to do that. That's what we're going to be learning about next semester, right? When we take statistics, we're going to be learning all about how we get all these results and how we're going to formally compare them and determine what are the magnitude of our effects and things like that. Now, this time, with and if that's really vague, that's okay. We're going to learn more about that in coming chapters when we start talking about descriptive and inferential statistics. For now, though, the small N design that relies a little bit less on analyses and a little bit less on tests and stuff like that. There they have what they call a visual inspection, and a visual inspection is basically where you look at a chart and you determine, based on the look of that chart, whether or not your independent variable had an effect. Very, very easy. You don't have to get math involved, but we'll talk more about that a little bit later in this video. And then finally, the goal of large subject designs is going to be to generalize to a population, whereas for here, we're going to be generalizing to a small group or even just to a handful of individuals. So previously, just now, I talked, in the previous slide, I talked about what happens if you are interested in studying somebody that has a neurological disability, an abnormality, anything that deviates from normal, or quote-unquote normal neurotypicality. So here is an example. This guy, his name is HM, and we've in this semester we have seen a video of someone like HM. Basically, HM's story, he's kind of this, he's considered the man without memory, or he was before he passed away. He lived quite a long time, but he died about a decade ago or so. HM is a quintessential example of a case study. So essentially what happened to him is that he had really bad epilepsy. He went in to have a surgery, and the neurosurgeons removed his hippocampus, believing that that was part of what was facilitating these seizures. And unfortunately, it was not the thing that was facilitating his seizures, and at this time we didn't really know what the hippocampus did. We didn't know. We knew it was a part of the brain and that it was important, probably, but we didn't know what kinds of behavior it managed. Now we do, because after his surgery, HM was unable to create new memories. So, and I keep calling him HM just before I go on. I keep calling him HM is because for a long time, a lot of results about his behavior and about his memory and about his neurology were published in scientific journals under the name HM to kind of provide him anonymity because he was still alive at the time. So just protecting his identity. Now, after he passed away, they released his name, which was Henry Molaison. So HM was unable to create new memories after losing his hippocampus. And the thing about this is that before now we had no idea what the hippocampus did. Nobody knew what it did. Now we can kind of, in a way, learn that, okay, if you lose your hippocampus, you lose all of your memory. That seems like a very clear causal connection, right? That essentially what that must mean is that something about memory is facilitated, is created by the hippocampus, because once you remove it, then it's gone, right? Then your memory's gone. And so through several decades, H.M. lived with, he lived in a kind of like a retirement, as an assisted living facility, where he worked with nurses and kind of needed help to live because he couldn't remember anything. He was constantly in kind of the mindset that he was going to have surgery later that day, even though it was decades later. So he wasn't able to remember a lot of things from his past. But he definitely wasn't able to create new memories. And mostly what he remembered was the only things that happened to him 30 seconds ago. After 30 seconds, you couldn't rely on him to remember any of that. It was just gone. So this is a rare neurological condition called anterograde amnesia. This is very rare. There's only, as far as I know, a handful of people who have really shown this to this kind of level of anterograde amnesia. So researchers spent a lot of time with him trying to understand how his memory works. Just to give you an idea, I'm going to go back to this slide. What you're seeing here, you see somebody drawing into the star. This is called a mirror tracing task. Essentially what this individual is doing is they are looking right here in this mirror and trying to figure out how to draw within the line of this star. It's really easy if you don't have a mirror, but if you have a mirror where every move you make is reversed, it gets a little tricky. It's hard at first. But here's the thing. By studying HM in close detail for a number of years, people were able to give him all kinds of memory tasks. And what they found is that for all the memory tasks, he showed no example of learning, no example of retaining memories. But on this task, he did show improvement. In other words, basically, he would struggle at first to get this right. And then after, a couple of days, he got really good at it. But he would never be able to tell you that he remembered doing this task ever in his life, but he would get better at it. Later on in his life, basically, they taught him how to ride a bike. He gradually learned how to ride a bike. But if you asked him, do you know how to ride a bike? He would say, no, I've never ridden one in my life before. And so that kind of systematic investigation of behavior kind of revealed a lot about memory. A lot of what we know about memory today is because of HM and because of the close systematic work of research with him while he was alive. And so if you're interested in that kind of thing, take cognitive psychology, where we talk about HM for a good chapter or so to talk about why it is he is able to do better at this, but not able to actually remember why or how he got better at it. So that's one example of a case study. There are other examples, though. Another example might be Ted Bundy. Ted Bundy, I think most of you probably know about him because he's notorious, but also because there was some Netflix, maybe a movie, maybe a documentary or a series or something like that came out not too long ago. Essentially, he's one of the kind of most widely known serial killers in America, and the reason why for that is because his acts were especially heinous. But I think people were especially interested because of how he presented himself in his normal everyday life. That basically he came across as a total, complete, normal dude. It didn't seem like there was any kind of malicious intent in his behavior whatsoever. There were no quote-unquote warning signs that would cause you to think that he would have done the things that he did. By most accounts, by people, he was a very charming guy who was very well-spoken. People thought that he might be a good politician, that kind of individual, And so after he was arrested, after he was busted, psychologists interviewed lots of people, not just him, but people that he knew, people like his neighbors, his family, his coworkers, people that knew him when he was growing up, and things like that. Because the answer or the questions they were wanted to answer is, why would he do this? Why would he do these really terrible, horrific things? And the reason why is because that kind of information is valuable. If we can look at things like warning signs for this kind of predatory, despicable, murderous behavior, that could potentially save people's lives, right? If we know the warning signs to look for, or to try to find ways to prevent that kind of thing from happening. So serial homicide or serial killing is not really a frequent occurrence. It doesn't really happen that much, and because of that, because it is rare, whenever people do it, people are generally interested in it, right? A lot of people, especially psych majors, are interested in that area of behavior because it is very unusual and it's very hard to understand why people would do those types of things. And because of that, there's this morbid curiosity there. All right, and so again, this has a huge impact on society. I'm mentioning here that you can't assign someone to a killer group. So this is an example of a study that, it has to be a case study because you can't directly manipulate the independent variable. You can't assign someone to be a serial killer and then see how that impacts their behavior. Another example, which if you have taken childhood development, you've definitely heard this name before, is Jean Piaget. And Piaget is one of the most influential psychologists of all time. But it's kind of interesting that he is, because I feel like he's not really talked about the same way, at least in my mind, as others are. So if I were to ask you to name some of the most influential psychologists, usually people will mention Freud. Freud was voted the third most influential psychologist of all time by psychologists. So he's third. You might even say John Watson, right, from the baby Albert studies. He ranks lower. You might say Pavlov. He ranks way lower. If you said B.F. Skinner is number one. B.F. Skinner is the number one most influential psychologist as voted on by psychologists. This guy, Jean Piaget, he's number two. So Jean Piaget is the second most influential psychologist of all time. So very, very widely influential. But here's the kind of interesting thing, is that he is very influential, and a lot of his methods kind of move beyond what we've talked about this semester. He didn't really do experiments most of his work wasn't really done with experiments the same way that we think of them in our class. Instead, he was more kind of involved with case studies, where he would take one or two, kids at a time and systematically observe them, make lots of notes about what they were doing, what kinds of behaviors they were interested in doing, what they believed, and things like that. And doing that really kind of close observation, he was able to generate lots of theories and lots of ideas about how development works. At what times do we learn about morals? At what times do we learn about object permanence? At what times do we learn about right and wrong? Well, I guess I already kind of said that. At what times are we able to think abstractly or read? Those types of things. And so, by getting all that data, he was able to make these theories, make these ideas, in which other people, sometimes people in his own laboratory, were able to test using, the typical tools of research methods, experiments, and things like that. And so his kind of designs were single subject in nature. If you go back and look at some of his original writings, it's interesting because he talks about one specific toddler instead of talking about, a group of toddlers like we've been reading in our research articles this semester. So in this case, his use of case studies was more about kind of exploring behavior and finding potential questions to help us understand how behavior works, in this case, how we develop. So there are three different kind of assumptions that go into single subject research or single N research. And ultimately, the three assumptions that the book outlines One is that it's important to focus on the behavior of individuals. In other words, the reason why that's important is because if you're looking at an individual, you don't have to worry about hiding all of that individual variability, all the individual differences, what makes us unique. You don't have to try to erase that anymore. You can focus on one person and see like exactly how it impacts that one individual, and you can see like how those individual differences matter. So let's say if you're left-handed, a lot of research with people are done with people who are right-handed, right? So if you are left-handed and this kind of research turns out differently, then maybe it has something to do with your left-handedness. That's just a really broad kind of oversimplification, but you get the idea that basically using the individual participant instead of a group tells us a lot about that individual, but it does tell us a little bit less about group differences or about how behavior works universally speaking than it does how much behavior works for one person. The second assumption then is going to be that it's important to discover causal relationships through the manipulation of an independent variable. That is not anything new. We've been doing that since chapter 5, which is all about experiments, right? We've been doing that kind of stuff for a while now. We know that it's important to manipulate the independent variable because doing that is going to tell us about cause and effect. More on that at the very end of this video. And then finally, it's important to study these kinds of effects that have biological or social importance, because if you're doing something like a behavioral intervention or a treatment plan for somebody, it's good to look at how this individual fares. It's not so good to maybe collapse across an entire group, because then if some all right, I'm gonna do a quick detour to give you an example of why that's important. So here we're saying a biological importance, right? So let's say that we're going to be developing a drug, and we're going to be developing, let's say that it is a drug that is going to help cure or help treat depression, just as an example. So let's say you test 100 people, you test them on and you test them off. And what you find is that when our people are on this antidepressant, that medication, that overall that people do or report being happier and less depressed. So okay, it seems like the medication works, right? But what this is asking us to do is to consider what happens for this individual. What happens for the individual who takes this kind of treatment? Maybe it's the case that out of that 100 people who did that, maybe 5 people really had really bad side effects. That would be covered up, not intentionally, but just by the sheer numbers of 95 people it worked, and for 5 people it didn't work, right? And so sometimes it's good to measure these on a person by person basis for things like that, things that could be dangerous, like side effects. All right, so here is an example of a single N design or a small N design. Here, this is from a study that is looking at dog phobia and specifically how to treat instances of dog phobia. And so in this case, what you're looking at here for these 3 different charts are going to be 3 different children that have that are further on the autism spectrum than most people. And so what you're looking at here is going to be, on the y-axis, how far away they are from the dogs and how far they want to be away from the dogs. And on the x-axis is going to be how many days have gone by, with each day being a separate treatment session. So basically this was to test whether or not this new kind of treatment plan was going to have these children come closer to the dogs. Pretty simple, pretty straightforward. So let's look at what happens for this individual right here, whose code name is John. This individual, you can see that he wants to keep his distance away from the dog, mostly 100 feet away, sometimes 25 feet away. Once they introduce this treatment plan, the first day, he's still not having it. But the second day, you can see that he immediately drops down. He's now very close to the dog. You get the idea. Now he's very close to the dog, and as the days goes on, he's still really close to the dog. He's not leaving the dog behind. And then Here, in the second piece, where the dashed lines are, we can see they introduce a new variable, which is that they are now introducing another dog. So it's 2 dogs instead of 1. Well, I'm going to talk about why that's important in just a moment. So here you can see a very clear cause and effect, essentially, right? Or what we think to be a cause and effect. That we see that this person has a phobia of dogs. There's this new treatment that takes place, and now he no longer has that phobia. Okay, awesome. Let's look at how it works for Sally. You can see that Sally, her phobia was not nearly as severe as John or as Bob's down here, but as soon as this treatment plan was introduced, you can see that it immediately drops. She's no longer afraid of the dog. And the same for Bob. That for Bob, he went on for quite a while, and then on looks like the 13th day or so, they introduced this experiment. This experimental therapy, and his phobia of the dogs pretty much went away, right? So, and the reason why this is kind of a cool design is because it is what is called an A-B design, where you have condition A, which is going to be our baseline, and condition B, which is going to be the treatment design, the treatment condition. So they have this baseline condition and the treatment condition, and you can directly compare and look to see whether or not there were any differences. And this is what I meant about the visual inspection part of this. We don't need to do complicated statistics to determine whether or not this had an effect. We can clearly see that it did have an effect on their behavior. So, the reason why you see here this treatment was established on like the 5th day, here on the 10th day, here on the 13th day, the reason why is because if you do this on the 5th day for everybody, and for everybody it decreased, we would not be able to say with certainty that the cause of their behavior, wasn't just the treatment plan, but it could also have been just after the 5th day, people naturally start to lose their fear. So to prevent that kind of interpretation or that possible confound, they did it at different times to make sure that that was not the case. Here, I said I was going to come back to this piece where they generalized across 2 dogs. This is called depending on your point of view, this could be an ABA design, or it could be an ABC design. Essentially here in this middle piece, we have this kind of therapy that they're doing, right? This treatment plan that is working towards getting kids to not be scared of their dogs. That's gone, and then they introduce another dog, and what they find is that they're totally fine with this new dog, right? And so in this case, we have basically a baseline treatment baseline plan. We're no longer doing that therapy. They're on their own, and we can see that it's still gone down, which means to us that this treatment must have worked and had some kind of long-lasting impact on their behavior. So that's a simple reversal design where you have a condition, you do a treatment, and then you do another condition that goes back to the baseline. So sometimes you'll hear this called ABA design. So baseline, treatment, baseline. Sometimes we hear it called an ABAB design. So condition, baseline, treatment, baseline, treatment. So the question in these cases is, does the behavior change? Does it change as a result of this therapy? And if it does change, can we be guaranteed that it will work and that it will last a long time? And so that's why we need things like going back to baseline. So let me show you an example of why that's important. So in this case, what we're looking at here is baseline and treatment. In this case, we are looking at OCD. So this is real data of an individual who has OCD. They're trying this treatment plan to see whether or not this treatment is going to have someone lessen the severity of their obsessions. In this case, I believe that this was somebody who was obsessed with germs. And so what we're looking at here is how many times per minute they have a thought about washing their hands. So in this case, you can see in baseline, before we introduce any kind of treatment, you see that it is pretty high, in some cases 13 times a minute, 9 times a minute. That's very, very frequent. They can understand how that would disrupt somebody's life. And then once the treatment is instilled, you can see that it drops down. In some cases, 0 times a minute, 1 time a minute, 2 times a minute, you get the idea. Then, just to make sure that it wasn't the effects of time or any other confound, they went back and got off of the treatment. And what they found is that once they got off the treatment, that rumination went back up. So you can see here for a baseline condition without the treatment that it goes back up. So they reinstate the treatment and then it goes back down. They take the treatment off again and you can see that it goes back up. They put the treatment back on and it goes down. They take treatment off, it goes back up, and you get the idea. So in this case, this is called an AB design. How do you know this? Because A is always the baseline, B is always the treatment, and so however many times you go back and forth, that's how you describe that particular design. So here is an example of something called an AB design, because there is no going back to baseline in this case. This is an example, so this is a study from the 80s that looked at the effects of memory training for people that have Alzheimer's. And so what you're looking at here is in the first 5 weeks, this individual was performing very, very poorly on a recall task where they are asked to recall a certain number of words. But after the treatment was reinstated, after the treatment was put into place, you can see that it goes up. And so the way that we interpret this is simply that once that treatment was kind of involved, you can see that memory goes up and it stays up. And for the researchers here, they had no real desire to go back to baseline because honestly for them, I think probably for them, they want to A, show that this worked, and B, they're not interested in going back to baseline, maybe for morality reasons, maybe it's impossible. If this were the effects of a surgery, you would probably not be able to go back to A. If this is something that is going to possibly hurt somebody's life, you can't go back to A. But for that reason, we should be kind of skeptical, right? Because this is just an ABA design. If we really wanted to discern causality here and to make sure that this really did kind of work, we would want to go back to baseline. So we'd want an ABA design. So in this case, this is a little bit more of a quasi-experimental design now because we are introducing an independent variable. In this case, it's going to be this new kind of treatment, and then they're not going off of it. So that's an example of an AB design. All right, so I want to very quickly talk, one of the last things I'm going to talk about here is going to be the validity in case studies. So we've talked a lot about validity this semester and why it's important and things like that. Sorry, I'm running low on battery, so I'll try to make this quick. So we talked a lot about validity and why it's important. So let's talk a little bit about for specifically case studies, why it's important, what we need to be thinking about. So remember that there are 3 different criteria for causality. That we have internal validity, we have directionality, and we have internal validity. Or sorry, I already said internal validity. We have covariance, right? And directionality can also be temporal precedence. So whenever we are doing these things, going back to this, we see the covariance. We can see that it definitely has an effect on that data. Does it have temporal precedence? Well, we're not 100% sure. We feel pretty good about it because we know that we directly decided when, that treatment came into place, but we're not 100% sure that if we were to get rid of it, that it would go back. And that's what we would need to make sure of for temporal precedence. And then finally, internal validity. Here, that's fine for this one. We developed a task that we thought was going to enhance memory. It seems like it does. We have no reason to believe that there would be anything wrong with that. Now, for validity, if we're specifically looking at validity now, for case studies, internal validity can often be very, very high because you are probably measuring what you think that you're measuring. You are able to look at individual differences. You are able to look at exactly how this individual is responding to this treatment plan or to this interview, and that's really helpful for internal validity. It's much better than it is in something like an experiment. External validity, on the other hand, is often usually worse than in an experiment. So remember, external validity or ecological validity is essentially how much does these set of findings generalize to other people. This is tough for case studies because oftentimes we may not always be interested in other people. So, for example, if we are talking about Usain Bolt, we're talking about the world's fastest person. We can't be 100% sure that the lessons that we draw from him about his training and his regimen and how he became the world's fastest person would be able to generalize to everybody. Maybe they don't. We don't know because we're only working with one person, and because we're working with one person, it's really hard to generalize to the rest of the population. So external validity usually quite a bit lower, and statistical validity is not really a factor so much in single subject designs or in case studies because you're not really using statistics. Instead, you're using that visual inspection method where you're looking at the data to see whether or not it had an effect instead of trying to use statistical tests to examine if there was or was not an effect. And speaking of those statistical tests, later in the semester, I think next week actually, we're going to be looking at those different kinds of statistics and what they can tell us about research, how we can use them to our advantage, and how we can basically get a bunch of data and find meaningful information in that wealth of data. All right, so that's all for now. I'll check back in with you later. Thanks for tuning in. Bye.